Okay, so our DNA is binded right now. DNA's helicase comes in and breaks this open. And DNA gyrase comes in to relieve any tension in the helix. So single-stranded binding proteins then come in to keep the hydrogen bonds separated, but this would be all over the place. And as you can see here, T has 2 hydrogen bonds, C and G have 3 hydrogen bonds. So then RNA primase brings in, on the leading strand, brings in RNA primer, which is right here. And DNA polymerase 3 binds the new strand one at a time. So it'd be T and A one at a time. And then our leading strand is finished. So then RNA primase brings in RNA primer, which is seen here. And DNA polymerase 3 again connects each letter by each letter, giving it its complements. And since this is known as the lagging strand, it has to work in 5' to 3' direction. So as the fork opens up, RNA primase comes in again and fills in an RNA primer. Then DNA polymerase 3 binds to the primer and then adds in the complements, each one at a time. So next you have DNA polymerase 1. All this does is it removes the RNA nucleotides and replaces them with DNA nucleotides. So for the leading strand, U, G, and A would be replaced by T, G, and A. And C, A, and U would be replaced by C, A, and T. And then A and G would be replaced by A and G. So next, the leading strand is finished, and now the lagging strand is not finished yet, as these are not connected. This is known as Ozaki fragments, and the phosphodiester bonds connect the fragments together, and then this is known as ligase, which is the glue, which is represented by the tape. So the tape, or the ligase, would basically connect these bonds together, and then you have DNA replication.